Hello everyone. Today we'll be solving Cambridge International ASN A level Biology Paper 5 Planning, Analysis and Evaluation May-June 2021 Paper 51. Question number 1. The enzyme beta-galactosidase catalyzes the breakdown of the compound ONPG. ONPG stands for O-nitrophenyl 1-D-galactopyranoside to the compound ONP. So basically ONPG is converted to the compound ONP, and the main responsible enzyme for that is beta-galactosidase, and along with it, it produces a galactose. ONP is a YOLO product. As ONP is produced, the color of the reaction mixture changes to yellow. The intensity of the yellow color produced is proportional to the concentration of ONP. So if there is more ONP, there will be greater intensity of yellow color. So a colorimeter is used to measure the absorbance of the reaction mixture. So basically, to follow the experiment, we can measure the absorbance of the reaction mixture. Absorbance is a measure of the light absorbed by a colored solution. All right. In the reaction shown, figure 1.1, the more intense the yellow color, the higher the absorbance. So if the yellow color is greater, which indicates there is a higher concentration of ONP, which in turn indicates that more amount of product has been produced. Part A. A student was provided with a stock solution of the enzyme beta-galactosidase. The student diluted this by a factor of 20 using a buffer solution of pH 8. The student made final volume of 10 cm³ of beta-galactosidase. So the student diluted it by a factor of 20 times, and the student made final volume of 10 cm³. Describe how the student prepared the 10 cm³ of diluted beta-galactosidase solution. So when our goal to reach is finally 10 cm³ after 20 times of dilution, so let's divide the 10 with 20, then we will end up with 0.5. So we will have to start with 0.5 cm³ of the original solution. So the student can take 0.5 cm³ of the stock solution and then add 9.5 cm³ of buffer in a volumetric flask, and that will make the total volume up to 10 cm³ of diluted beta-galactosidase solution. The student investigated the effect of substrate concentration on the enzyme catalyzed reaction shown in figure 1.1. The student was provided with the diluted beta-galactosidase solution prepared in step A, which was kept cold until needed, a stock solution 1.0% ONPG made up in a buffer solution of pH 8, and a buffer solution of pH 8. The procedure used by the student is outlined in step 1 to step 3. The diluted beta-galactosidase solution was mixed with 1% ONPG solution. After 2 minutes, a colorimeter was used to measure the absorbance of this mixture. Step 1 and 2 were repeated using different concentration of ONPG solution. The question says, suggest why the student used a colorimeter to measure the absorbance rather than judging the intensity of color by eye. So when judging any color by eye, we have to understand that we have difficulty judging color, specifically when a particular color changes, our endpoint is reached. We have difficulty, and if we use a colorimeter, the results are going to be quantitative. Part 2. Identify the independent variable and the dependent variable in this investigation. First of all, if we talk about the independent variable, whenever we are asked about the independent variable, we have to understand independent variable is the variable that we are allowed to change, and dependent variable are usually the answer, usually the result of that particular change, and controlled variable are those that we will keep fixed so that their change in the controlled variables change in parameter does not affect the reaction. So in this case, the independent variable is actually the concentration of the ONPG, or the substrate that we are using. And the dependent variable is the absorbance on the colorimeter, how much absorbance is occurring in the colorimeter. The student used the absorbance values at 2 minutes as the initial rates of reaction. Figure 1.2 shows the result. Initial rate of reaction, percentage concentration of ONPG. So we can see as the percentage concentration of ONPG increases, the initial rate of the reaction increases, but at a certain level of ONPG, we can see that the initial rate of reaction is no longer increasing. That means substrate concentration at this particular point is no longer the limiting factor. Part C. The student decided to investigate the effect of inhibitors on the reaction shown in figure 1.1. The student planned to add an inhibitor. The inhibitor is X to the reaction mixture containing different concentrations of ONPG solution. Describe a method that the student could use to collect the data needed to test the effect of inhibitor X in reaction mixture containing different concentrations of ONPG solution. The description of your method should be set out in logical way and detailed enough for another person to follow. You should not repeat the details from A describing how to dilute the stock solutions of beta-galactosidase. Okay. When performing an experiment like this, first of all, because the experiment tells us that we have to prepare different concentrations of ONPG solution. So we must prepare different concentrations of ONPG solution. At least 5 different concentrations must be prepared, and we must mention that what percentage of concentration that we are using. All right. And another thing that we must also follow is that in this particular experiment, we already know that the diluted beta-galactosidase, the enzyme, was kept cold, all right, until it was needed. So thereby there must be a method of keeping that particular beta-galactosidase cool. All right. And because our experiment is going to take place at room temperature, so before the experiment, both the enzyme and the substrate must be brought back to the room temperature before doing the experiment. So we basically store the enzyme at a lower temperature only to keep the quality of that particular enzyme. And the stock solution, which is 1% of ONPG, we can only make more dilute from here. So 1%, and then we can go like 0.8, 0.6, 0.4, 0.2, and we can go like that kind of percentage. With the buffer solution, we can keep on adding buffer solution with the stock solution. So basically, that's how we can make more dilute. And a buffer solution of pH 8, so as to keep the pH constant. So guys, to describe a method that the student could use to collect the data needed to test the effect of inhibitor X in a reaction mixture containing different concentration of ONPG, what we have to do is we have to first prepare 5 different concentrations of ONPG from the stock solution. So that we had ONPG stock solution of 1%. So we have to prepare like 1%, 0.8, 0.6, 0.4 and 0.2. The beta-galactosidase enzyme which is used can be kept cold using cold water bath before the experiment, and right before the experiment, each experiment, the enzyme and the substrate temperature can be equilibrated before mixing. A thermostatically controlled water bath can be used to keep the reaction mixture at 37. If we are doing the reaction at 37, the experiment maybe can be done at 30 degrees Celsius as well. So whatever is the case, a thermostatically controlled water bath will do the job of maintaining the temperature. Same volume of inhibitor was used for all experiment. So for all experiment, same volume of ONPG substrate. However, not the concentration, the volume of ONPG substrate was used for all experiment. Same volume of the beta-galactosidase enzyme was used for all experiment. The intensity of the yellow color can be measured after 1 minute for all experiment. So it's like a standard. For all experiment, after 1 minute, we're going to measure the intensity of the yellow color. For each concentration of substrate, the experiment was repeated at least 3 times so that we get an average. All right, to get an average. Beta-galactosidase may cause allergy in some people, so gloves must be used as a precaution. Figure 1.3 shows the results when no inhibitor X was added. Percentage concentration of ONPG, initial rate of reaction, it increases and then it plateaus. There was no inhibitor X. The student suggested that inhibitor X was acting as a competitive inhibitor. On figure 1.3, sketch the curve expected if inhibitor X was acting as a competitive inhibitor. A competitive inhibitor does not stop the reaction from taking place. It just occupies the active site for as long as the concentration of the substrate is not enough. So when the concentration of substrate can go beyond a certain point, then it can knock off that particular competitive inhibitor from the active site and the reaction still take place. So all we need is a higher concentration of ONPG and we will be able to get our desired initial rate of reaction. So our graph is probably going to look like this. We're going to meet it at a higher concentration of ONPG. Vmax is the maximum initial rate of reaction of the enzyme. The Michaelis-Menten constant Km. The Michaelis-Menten constant Km is the substrate concentration at which the initial rate of reaction is half its maximum value, which is Vmax. Now the question says, draw on figure 1.3 the position of Vmax and Km of the enzyme when no inhibitor X is present. So basically we have to understand the maximum velocity at which a particular enzyme works is its Vmax. So we can see the enzyme is plateauing at this particular location. This is the enzyme that is plateauing. So the Vmax should be that, and half of that, so we can see half of that is going to be here. So this is Km, which is the Km substrate concentration. This is Vmax, and this is half Vmax. Use your graph to describe the effect of the addition of inhibitor X on the Km of this enzyme. So we can definitely see the addition of that inhibitor increases the value of the Km. We can definitely see it increases the value of the Km. So this is the new Km in the presence of inhibitor X. So we can say the reaction with competitive inhibitor has increased the Km value. Part D. Acid reflux is a condition where some of the stomach contents are forced back up into the esophagus or gullet. The main symptom is a burning pain in the esophagus due to the acidic contents of the stomach. Acid reflux that happens more than twice a week is called gastroesophageal reflux disease or GERD. Two main types of drug are used to treat GERD: proton pump inhibitors (PPIs) and H2 antagonist (H2RAS). Scientists carried out trials to investigate the effect of these drugs on the relief of acid reflux symptoms in people suffering with GERD. 200 people were randomly divided into two groups, A and B. People in group A were given PPI medication. People in group B were given H2RA medication. The trial lasted for 16 weeks. In week 4, 8, 12, 16, the people were asked to score their symptoms using a scale shown in table 1.1. So we can see there is a score of none, minimal, mild, moderate, moderately severe, severe, very severe. Mean values for each of the treatments A and B were calculated for weeks 4, 8, and 12 and 16. People with a score of 1 was classified as showing removal of symptoms. People with a score of 2 or 3 classed as showing improvement of symptoms. The results of these trials are shown in figure 1.4 and 1.5. So removal of symptoms. Weeks after starting treatment, mean number of people reporting removal of symptoms. Okay, PPI, H2RA, error bars, 95% with CI. So we can see that removal of symptoms with PPI, alright, we can see there is an increase in removal of symptoms. Initially there is a large increase, but then later on the removal of symptoms, they do not report as much. However, with H2RA, over the course of duration, the removal of symptoms is higher. Improvement of symptoms. Okay, we can see mean number of people reporting improvement of symptoms. With 4 weeks of treatment, we can see there is a huge amount of improvement with people that are taking PPI. But prolonged duration of taking it does not increase it significantly. However, with H2RA, we can see that with 4 weeks there is not much of an improvement. However, prolonged taking in the medication does improve the condition significantly. We have to understand also another thing: with PPI, the error bars are overlapping. So it may happen that the person may not have any further improvement after 4 weeks. Scientists analyzed the data and concluded that PPI should be used to treat acid reflux rather than H2RAs. Alright, with reference to the data in figure 1.4, figure 1.5 discuss the conclusion that PPI should be used to treat acid reflux. Well, we can say PPI has a huge advantage within the first 4 weeks, which the H2RA doesn't have. Guys, the scientists analyzed the data and concluded that PPI should be used to treat acid reflux rather than H2RS. With reference to the data in figure 1.4 and 1.5, discuss the conclusion that PPI should be used to treat acid reflux. Now, whenever we are asked about a question that says discuss, then we must write both for and against if possible. So first of all, we can see that if we are using PPI, we can see there is an immediate improvement. There is an immediate improvement of symptoms. There is an immediate removal of symptoms, which is a great thing. All right. I mean, everyone wants to be cured from their disease. So at 12 to 16 weeks, however, we can see there is an overlap of the error bars, means the difference between PPI and H2RA is not significant. That means over the course of a long run, maybe H2RA could be a better effective medicine. All right. Then moving on. There is no statistical test to compare the groups. So yes, obviously we can see that both of these groups, there were no t-test or any kind of test that were done. So we can actually tell the significance of this particular data. Then another thing that we can comment on is that there is no information on severity of symptoms before trial. And severity of a particular symptoms can be subjective from person to person. Pain is subjective from person to person. So that there can be an error in that particular part. There is no data on long-term effect where H2RA may be better. Even though we can see that H2RA is improving. H2RA is improving the symptoms, improving the symptoms. However, there is no long-term data, all right, that whether H2RA will be better or PPI will be better. So keeping that in mind, there is also no data on the side effects. So difficult to tell why one type of treatment will be chosen over the other one. Question number 2. Inheritance of flower color and flower position in pea plants are controlled by two genes. The gene capital P and small p controls flower color. Allele P for purple flower is dominant to allele small p for the white flower. Capital A controls the flower position. Capital A for flower growing from the side of the shoot. Axial position is dominant to small a for flower growing at the end of the shoot. So at the side, which is capital A, and end of the shoot, which is small a. All right, and then capital P for purple color and small p for white flower. Okay, so a biologist predicted that if the genes are on different chromosomes, the ratio of the phenotypes of the F2 generation would be 9:3:3:1. Well, the biologist is correct that it should be that. The biologist carried out a breeding experiment. Plant homozygous for white flowers and axial positions were crossed with plants homozygous for purple flowers and axial position. All the F1 had purple axial flowers. The F1 were crossed with each other. Table 2.1 shows the results for the F2 generation. So in F2 generation, we can see purple axial flowers 1756, purple terminal flowers 653, and we can see the whole point. Okay, the chi-square test was used to analyze the data in table 2.1. State one reason why the chi-square test was used. So a chi-square test is used when we want to compare between observed and expected data. All right, and the data is categoric and discontinuous. So for that reason, a chi-square test would be a best test for this particular purpose. Part 2. State the null hypothesis that the biologist would use for this test. So a null hypothesis would be that there is no significant difference between the observed and the expected result. If there is any difference, then it would be due to chance. Part 3. Complete table 2.2 and calculate the value of chi-square for the results of the F2 generation. The equation for the calculation is chi-square, sum of observed minus expected whole square divided by expected. Now, purple EDEXCEL flowers. So in terms of ratio, we are supposed to get 9:3:3:1. So if we divide 3,345 with 9:3:3:1, which is equals to 16, then we get a total of 209.06. Okay, 209.06. Okay, so the observed will be, the expected should be, since it is a ratio of 9, so the expected should be multiplied by 9, 181.56. So observed minus expected will be 125. Observed minus expected whole square, so we will have to square it, divided by expected, which is, it gives us 8.379. So 8.37, or yeah. So for the observed of purple terminal flower is 3 ratio. So we'll have to repeat the math. 627.18, 627.18, and the 1 ratio will be 209.06. Now in every cases we'll have to do observed minus expected. So let me show you a demo of how we are doing it. So observed is 653 minus expected, which is 627.18, and then we'll have to square the value divided by expected, which is 653, and that's how we are getting this particular answer. So the value is 1.062. Okay, the next value is 8.92, and then the next value is 2.99. Chi-square is equals to 21.48. Now table 2.3 shows some critical values of chi-square at different probability levels. The degree of freedom and the probability is given. Our exam probability is always 0.05, and the degree of freedom is always, since there are 4 terms, 1, 2, 3, 4, the degree of freedom will be 4 minus 1. So the degree of freedom will be 4 minus 1, that means 3. So with 3 and 0.05, our value should be maximum 7.82. State the critical value probability less than 0.05 for this chi-square test. Well, the critical value is 7.82. Now the question says use your calculated value of chi-square to explain whether the null hypothesis should be accepted or rejected. You see, if our value would be lower than 7.82, we would accept the null hypothesis that there is no significant difference. Since our value for the chi-square is 21.48, so our difference is significant. So we are not going to accept the null hypothesis. We're going to say that the null hypothesis is rejected, and chi-square is higher than 7.82 or the critical value. So we will also say that there is a significant difference between the observed and expected result. suggest a conclusion that the biologists could make about the inheritance of the genes controlling flower color and flower positions in pea plants. So the scientist, because we are rejecting the null hypothesis, the scientist can say that the genes are on the same chromosome. All right, so they are autosomally linked and does not assort independently. The biologist Guys, thank you for joining the video. We are going to upload videos like this, related to chemistry and biology question paper all the time. So please subscribe to our channel and stay with the channel, and let us know your feedbacks in the comment section. If you want any particular question paper solved videos, if you need it like really fast, let me know in the comments. I do read all the comments, I check them, so I will definitely try my best as soon as possible to upload your requirements. Thank you for joining the video. See you in the next video. Bye bye.